Today, we're announcing the launch of the CNBC Cures initiative, focusing on advancing awareness and understanding of rare diseases. My 9-year-old daughter, Kaylee, has a rare genetic condition called SYNGAP1. It's a brain protein deficiency that results in autism, seizures, developmental delays, and there are less than 2,000 other people in the world like her. The patient populations for rare diseases are small, but there are more than 10,000 conditions that you'd call rare. Altogether, it impacts some 30 million Americans. Many of those patients are children, and 95% of these diagnoses don't have an FDA-approved treatment. Despite those eye-popping numbers, millions of people continue to suffer from diseases that often go overlooked by the broader healthcare industry. Small patient populations mean that drug companies and investors may not be willing to invest in the space, and outdated regulatory frameworks make the process of developing life-saving treatments too slow for the families that need them. Joining us right now is one of the first people that I turned to for advice and guidance when we first started to go down the path of being rare disease parents. CNBC contributor Scott Gottlieb. He is the former commissioner of the FDA, and he serves on the boards of Illumina, Pfizer, and United Health. He's also on the CNBC Cures Advisory Board. And Dr. Gottlieb, thank you for being here.
Thanks a lot. Thanks for having me.
You were one of the first people I talked to because I just wondered what was possible, what was out there, what might happen. And you told me about some pretty exciting and amazing things that were taking place in the world of research and things that the FDA was doing to try and help push those developments along. Why don't you talk a little bit about what you first learned at the FDA and how that kind of changed what the FDA was doing at that point?
Yeah, and I remember our conversation. At the time, we were starting to think about what we were calling these N-of-1 diseases, and this was really galvanized by a single parent, a woman by the name of Julia Vitarello, and a doctor at Boston Children's, Timothy Yu. Her daughter had a rare disease, Batten's disease, but a very specific mutation. And what the doctor was proposing to do was to fashion an siRNA oligonucleotide. I heard you mention that in the last hour. That would be very specific to the particular mutation that this little girl had. And it could potentially be a treatment. And they wanted a pathway to take this forward. We ultimately created that pathway for them. They were able to treat her daughter. Her daughter did derive some benefit from that treatment. She unfortunately passed away during COVID, but it started to change the agency's thinking about how to create a pathway for these so-called N-of-1 diseases, where you might want to fashion a very specific construct for a very specific genetic mutation, and how could we operationalize this into a policy construct. And this has continued. We made some progress when I was there. You've heard FDA recently talk about trying to advance these frameworks. This was ultimately adopted into forms of policy. I think it was personified in what the president tried to do with Right to try to get these treatments out to patients more quickly, these gene therapies. And the technology has really advanced since then. There's much better constructs for trying to treat these diseases. And ultimately what the agency is going to need to do is to start to think about new drug therapies for these ultra-rare diseases, not as single products, but regulating the process that is used to derive the products rather than the product itself. And so if you follow...
What's that mean?
Well, if you have a genetic disease that's has a sort of common phenotype, and you know that the disease is being driven, for example, by the production of abnormal proteins or a missing enzyme in the brain, which a lot of these diseases are. If you can design a construct to address that specific mutation, that's going to have the effects that you're intending for, so reduction, for example, in the production of a certain aberrant protein, you should be able to get approval just on the basis of that construct itself, if you can prove that the oligonucleotide or the other construct that you're using is going to have that biological effect. So rather than having to do a clinical trial on every individual patient, because in many cases you can't find enough patients to enroll in a clinical trial and power it adequately, what you're doing is regulating the process by which you're trying to derive a construct that would have an effect in that biological system.
What is happening on the scientific front that you described, even the things that were happening in 2017, that those are outdated at this point? We're looking at much different ways of pushing forward.
Yeah, that's right. There's been a lot of progress just in those intervening years. I mean, what really opened up the field of gene therapy was the development of the AAV vector, a viral vector that could deliver genetic changes to people who were born with genetic diseases and other patients as well. The first time that we used an AAV vector in a patient was really the late 1990s for the attempt to treat cystic fibrosis. And so this is relatively new technology. And there's a lot of shortcomings of the AAV vector, including systemic side effects that you get from it. If you try to deliver it systemically, it oftentimes localizes in the liver. and create certain side effects that could be very dangerous. And we've seen
Or
kids
even
die in
deadly.
clinical trials.
Yeah.
Right, exactly. And so we've seen new technologies like lipid nanoparticles where you encase the genetic change that you're trying to deliver in a lipid nanoparticle directly to the target tissue, and you can target it to specific kinds of body tissue. We've seen things like synthetic capsids where you're, again, encasing the genetic alteration inside a capsid rather than inside a viral vector, rather than relying on the virus to deliver the gene into new tissue. You're able to more directly target it to that tissue. And so these are relatively new advances. They're in clinical development. They've shown good results in clinical trials. Companies like Beam and Verve are also doing things like base editing, where rather than delivering a whole gene, what you're doing is delivering a particle that will just modify the gene that's already in a person to try to make it a normal gene. And so we're having very good success with this technology.
It's somatic cells, though. How do you do it so that it would be, I mean, in other words, you can't get it into the stem cells where, does it stay in a somatic cell long enough to be expressed and provide the protein?
Yeah, I mean, that's the critical question. So you're not getting into gametes. You're not trying to produce a change that's going to be inherited. What you're trying to do is alter tissue that's in a patient's body.
And it can stay there and do it for 6 months?
It could stay there and do it potentially for years and maybe in perpetuity. What we don't know is the durability of a lot of these treatments and whether or not patients are going to need to be redosed with these constructs. And that's also the problem with the AAV vector, because many times you can't redose a patient because they develop neutralizing antibodies.
Yeah, it's a one and done if you're sitting on
associated adeno.
Right.
Yeah.
So with these other constructs, presumably you can redose patients if the genetic alteration dissipates over time.
Wouldn't it be nice just to make the protein and introduce it into the, you can't do
We're doing that. Well, we are doing that.
But it's hard. What happens? It gets broken down?
It's broken down. You develop neutralizing antibodies.
Pass the blood-brain barrier.
Can't get it into the brain. You need constant infusions. Patients develop neutralizing antibodies.
It's better to have it done this way.
Even with ASO, you'd have to have it done every 3 to 6 months. It fades away, and it's not an easy injection. It has to go in.
Intrathecal.
Yeah.
Look, we've transformed diseases like Gaucher's disease with these infused enzymes, but if you can deliver a genetic alteration that provides a more durable treatment safely and comparable, if not better, efficacy, that could potentially be preferable.
The messenger RNA vaccines were delivered in a little lipid.
They were delivered in a lipid nanoparticle.
Is that where the technology
That's where the technology
comes from?
really has been perfected, yeah.
Scott, what's the biggest roadblock, I would say, and then the biggest hope you have on the other side of that?
Yeah, look, I think the technology is really promising, so I wouldn't say anymore that the technology is the biggest roadblock. I think regulation is a roadblock here because it's still hard to do these so-called N-of-1 trials. I think while the FDA has opened up a pathway to this, and we're able to do it in this particular case for this particular child and others since then, the FDA hasn't created sort of a reproducible framework. They put out a paper in November talking about this plausible mechanism pathway that they were going to create that would operationalize this at a larger scale.
Right.
I think ultimately
we had Marty
Congress...
carry on to talk about that.
Right. I think Congress should intervene here. I think there's room for legislation that would give the FDA very targeted authorities and resources to do this.
Did you just have a lot of continuing education, I think. You're an MD, right?
Yes.
You just read about molecular biology constantly, or something?
You're a biochemist.
Well, I know, but when you talk, MDs can't talk the talk like you do. They don't know any of these things you're talking about.
I think a lot do, and I think certainly the people at FDA are very well versed in this.
Because they've kept
They're
up
working
with the
on
literature.
it. Look,
yeah.
when I was there, we put out a statement that we thought there were going to be upwards of 10 cell and gene therapy approvals a year, and at the time that we said that, people thought it was absurd. We're close to that. We've had 7 approvals in the last 2 years. Last year it was down. It was only 5 approvals. But I think we're getting to a run rate where these things are becoming more mainstream therapies.
I mean, you're amazing, Scott.
Yeah,
People want, yeah.
And we are very grateful to have you on the board with CNBC Cures.
Thank you.
Scott Gottlieb.
You'd be good at the FDA.
Thank you.
Oh, wait.
It's done that. All right. For more on my family's journey with rare disease, you can check out cnbc.com/cures. And please join us for an in-person Cures event that's happening later this year in New York, March 3rd. You can click there to sign up for it. We're going to be bringing together some of the best minds to try and make a difference. Go to cnbcevents.com/cures to register for it. Squawk Box will be right back.